The Wisconsin Center of Excellence in Genomics Science.
Wisconsin CEGS.
The CEGS project. That Wisconsin CEGS project.
The Center for Excellence in Genomics Science.
The Wisconsin CEGS is a multidisciplinary research effort with researchers at the Medical College of Wisconsin, the University of Wisconsin and Madison, and Marquette University, and is sponsored by the National Human Genome Research Institute of the NIH.
The CEGS project is really very new and interesting because it's coming up with new technology to look at the genome in a different way and identify all of the proteins that are bound to all of the different regions in the DNA genome of different organisms.
So what's the challenge here? We currently have methods that allow us to investigate individual proteins and study where they bind throughout the genome. But we don't really have ways of investigating a particular sequence and characterize all the proteins that bind there and how that changes when a cell is challenged or in a disease state.
So you can observe that different genes are turned on and off, maybe in different levels and maybe respond to different things, but you don't know why. We don't have the tools right now to understand how that's accomplished. So this project, and the reason it's exciting, is a project to develop a new technology that'll allow us to peer into the genome and find out what proteins and other molecules are where in the genome, what genes are they working with, and be able to observe all of that changing with time and being different in different cell types and responding to external stimuli.
Methods have been developed that allow us to characterize and analyze proteins in a high throughput manner and highly parallel. This method, called proteomics, basically doesn't investigate an individual protein any longer, but you look at all the proteins of a cell simultaneously.
I am a protein biochemist by my education, and so I actually come in at the front end of the project, and it's my job to isolate proteins from those specific regions of the DNA and pass them on to the other chemists here in the group so that they can do the protein identification.
We're basically trying to reverse the DNA chip process, and in doing so, the final analysis requires mass spectrometry. And so my background is as chemistry and as a mass spectrometrist. So when we ultimately want to analyze our samples, we want to put them into the mass spec and run the data and do the analysis, and that's kind of where my end comes in.
Our goal is to be able to do sequence-specific capture, in other words, based on a particular gene sequence that we know, because we know the human genome sequence. We want to be able to go in and fish out that segment of the genome and also pull out with it the proteins that are interacting with that segment.
So how would this method actually work? Well, what we envision is that you would take cells at any given time point. You may be interested in them because you've challenged them with a particular toxin or they're at a specific disease state. And as a first step, you would basically take a snapshot of how all the proteins are binding to DNA. So you would cross-link chemically all the proteins to the DNA in the first step, and then in the next step, you would basically break up your cells. You'd take that molecule of DNA with all the proteins attached, and you'd break it into smaller pieces that are more easily to handle using standard molecular biology technology.
We need to bring together surface chemistry, because the way we're going to fish out these sections is by having surfaces that have gene sequences on them that we put there, and then using those gene sequences to capture the complementary gene sequences that we're interested in. So surface chemistry. Once we get the chemistry working and the molecular biology working, we also then need a very powerful analytical tool called a mass spectrometer, which will allow us to identify and eventually quantify what proteins are present on those minute amounts of material.
So the last challenge then for this entire process is that you get a lot of data, but you need to develop the appropriate tools to be able to look at these and analyze them computationally and integrate them with additional data that you have on the physiology of these cells or on the biology behind the system that you're studying. And so the last part of the CEGS grant is focused on developing efficient tools to facilitate this type of analysis.
This is a hugely collaborative project. We have surface chemistry, people who are experts in synthesis. You have your molecular biology people, you have your DNA people, you have experts in proteomics from the discovery side and the targeted side. So it's just a lot of people working together for a similar cause. And when you're forced to do that, you end up learning a lot.
It has a collaboration with University of Wisconsin-Madison and Marquette. And going there and doing some of these experiments and getting some of the knowledge about all these other different techniques and meeting some of the other great people that are working on the project has really opened my eyes, because you try to be specific and focused in one area, but you have to venture out.
Its impact initially will be for researchers and biology in general, gaining insight into what makes humans and other organisms work.
We're going to be able to use this technology to understand biology, both to learn how organisms function, but also to understand ultimately human health and human disease. So the more we learn about what normal cells look like, the more we'll be able to recognize what defective cells look like when there's a mutation or a disease such as cancer. And if we can understand why there are problems in the cell that cause these diseases, we'll have a better tactic to go in and try to bring new treatments and preventative medicine for human health.
So there's clearly a great need for this type of technology, and a seemingly unending number of questions, biological and medical, that we can ask and that we can apply this technology to once we have it.